At the Future Kings Biomedical Sciences program, students learn cutting-edge biological techniques. Our days start at the Discovery Center located in Manassas, Virginia. Sessions usually begin with presentations about a specific aspect of biology. Once students become acclimated to the topics, they then can present their own findings to a new generation of Future Kings. After presentations and discussions on the material, the group walks to the laboratory portion of the Discovery Center. The rest of the day is spent in a fully functional clinical genomics laboratory. Because safety is of the utmost importance at Future Kings, students are given lab coats and sterile gloves to work with. In the lab, students practice basic skills like pipetting, which allows for the sterile transfer of liquid, and vortexing, which allows a person to mix two different substances. Students are also exposed to cell culturing, which is the process by which cells are grown under controlled conditions, generally outside their natural environment, like a human body. The cells live in the red media, which provides nutrients for their growth. The lab also contains a compound microscope, which can be used to view microscopic organisms. The group works with many types of cell lines, like these HEC293 cells. Biomedical science students also have access to a biological safety cabinet, which is a device that allows a researcher to work with potentially hazardous materials without being exposed to them. Students have also been able to process COVID PCR tests on themselves using this equipment. After processing the saliva samples in the biological safety hood, the students use the state-of-the-art Quantstudio 7 Pro to determine if any COVID-19 virus was present in the saliva samples. Saliva samples were added to the small plate that is placed inside the Quantstudio 7 Pro machine. These samples are combined with a PCR mixture that contains various enzymes and compounds that allow a researcher to generate more copies of whatever RNA is present in the sample. The machine then uses lasers and a camera to record how many copies are made over time. Once the PCR is completed, the students can see which RNA was amplified. One line on the graph indicates that only human RNA was amplified. This is considered a negative test. Two lines indicate that human and COVID-19 RNA was found in the sample. This is considered a positive test. Future Kings biomedical students also have access to an ultra-cold -80 degrees Celsius freezer, which is used to freeze samples to the point where biological processes are virtually halted. Here we store samples for later testing and analysis. Illumina, an industry leader in biological assays, also kindly donated an iSeq 100 system that can quickly sequence DNA and RNA of interest. The biomedical students are also exposed to the biological techniques of the future. In this program, they learn to code and operate liquid handling robots. The life science robot company, Opentrons, readily donated an Opentrons 2 robot to the program. These machines can quickly and efficiently move liquid or samples between wells and plates. These machines are oftentimes better than humans at pipetting in a consistent and repeatable manner. In the Future Kings biomedical program, we strive to motivate students towards careers in biomedicine or biotechnology. We provide students with a competitive advantage to enter college with these biology skills, experiences, and exposures that the colleges will find attractive. Ultimately, we are grateful to have the opportunity to invest in these young minds that will lead the future.